So when people started to realize in the early part of the 1900s that Mendelian genetic existed and that it worked, then one of the things they started to do is look at all kinds of organisms and all kinds of characteristics to see whether Mendelian genetics was this grand universal answer to everything they ever wanted to know about genetics. And it turns out that when you come up with a really nice scheme, as Mendel did, and it appears to be working very well, it generally doesn't take very long until somebody finds something that they don't understand. Mendelian genetics, there it is, and this is how it's supposed to work, and here's my model organism, and it just doesn't seem to work at all. And so this next part deals with some of these perhaps anomalies where Mendelian genetics maybe doesn't give us all the answers. One of the questions when it comes to these universal answers to what happens in biology is that there are sometimes patterns that maybe don't perfectly work with what Mendel predicted, but maybe it's not that Mendel was wrong. Maybe we shouldn't throw out the whole idea. Maybe we should just look very carefully at how this new phenomenon works and then compare it in great detail with Mendelian genetics. There are these special cases. In fact, there are 4 of them that have to do with the genetics and one additional one that has not something to do with genetics. And these 4 are incomplete dominance, multi-allelic inheritance, Pleiotropy, where a single gene affects several aspects of an individual, and the reverse of this, polygenic inheritance, where multiple genes affect a single trait. And so these 4 are things people identified, and what we're going to do is go through each of them with some examples to make you understand better how ultimately Mendelian genetics still wins out, but we just sometimes have to understand what exactly the condition is. So the first of these is called incomplete dominance. And in incomplete dominance, one of the issues is that the offspring don't have the phenotype of one of the parents, but they have a blended phenotype. Blended phenotype is not what we saw with the pea plants. There we had 100% of the pea plants be purple flowered, and the white just sort of disappeared for a bit, and then it came back in the second generation. So here we have this blending. Now the blending does not mean Mendel was wrong. It simply means that the particular aspect of how color is produced is a little bit different. Mendelian genetics still works, as you'll see. It just has something to do with the red and white color, in snapdragons in particular. So if we set this up just like Mendelian genetics, and we start with the pure breeding red and the pure breeding white snapdragons, and then we form the gametes and we create the first filial generation, then you will see that they are all, 100% of them, pink. So the business about 100%, that's still Mendelian. So it tells us that some of this still works. It's just that the color doesn't work. Instead of having either the red or the white, the dominant one, emerge as the single color, we have a mixture. We have pink. So what this means is that the red does not completely dominate the white. So white is still recessive, red is still dominant, but it's incomplete. So let's just accept that pink exists and that pink is the heterozygous version of this. And so this is an incomplete dominance. In incomplete dominance is the case where you have 2 alleles, a red allele and a white allele, produce 3 colors, red, white, and pink. But pink is when the 2 are mixed, so that's only in the heterozygous ones. And then you can confirm this by producing the F2 generation. You take your pink flowers and you allow them to interbreed. And when you do this, there's only so many ways you can create eggs and only so many ways you can create sperm. You use this whole thing as a multiplication table and ta-da, this is the second generation. And the second generation has these numbers quite predictably, so Mendelian genetics still works. It's just that the flower color doesn't quite work. And so rather than saying Mendelian genetics is wrong, what we need to do is accept that there are certain traits that become blended. Not all traits do this. Clearly the flower color in peas didn't, but the flower color in snapdragons does. And there are all kinds of plants where you'll see that have two different flower colors, but there is incomplete dominance when it comes to color, and this incomplete dominance just happens. There are also other conditions, this is perhaps more relevant to us, where this kind of incomplete dominance happens with some of our traits. There is a disease called hypercholesterolemia, and hypercholesterolemia means that your cholesterol is not properly mopped up by your cells. Now normally your cells will have these so-called LDL receptors. LDL is one of the kinds of cholesterols. We all need cholesterol because it is important for our cell membranes. It is something we use to make hormones, but we only need a certain amount. And whatever we have in excess, we should have some way to remove it from the blood, because if we don't, then of course it can form plaques and obstruct our blood vessels and so on and so forth. So the normal cell exists in individuals that are homozygous dominant for the gene that makes LDL receptors. So this is the normal condition. If you look at the heterozygous individual, the heterozygous individual only has one functioning allele, the big H. That is the one that makes this LDL receptor, whereas the little h doesn't. So now if you are heterozygous, that means you make some of these receptors, but you only make half of them. And half of them means you have a mild case of hypercholesterolemia, which means you have to be careful what you eat. You can mediate this by having a low cholesterol diet and you'll probably be fine. But if you have both of the recessive alleles, so this would be your little h, that means your body cannot make any of these LDL receptors, so therefore they cannot mop up anything, and in addition to being careful with your diet, you really have to have something that removes the cholesterol from your bloodstream for you. Otherwise, you're always going to have high cholesterol. And so this is an indication where you have the same type of thing that you had with the flower colors and the snapdragons, but here it really matters. I mean, this is a real health issue. You need to have something ideally that helps you mop up these the additional LDL cholesterol. So that's incomplete dominance. Now the second of these is a situation where we have multiple alleles, and in addition we have something here called codominance. And the example we use here is the ABO blood group, which we have. Now the ABO blood group gene produces 4 blood types. So clearly there is something extra going on, because what we had in the past is we had 2 alleles and they produce 2 colors. That was the strict Mendelian genetics. Then we had 2 alleles and they produce 3 colors. That was incomplete dominance. And now we have something different. We actually have 3 alleles and they produce 4 colors. So not just 2 alleles, but multiple alleles. And how does this work? Let's start with blood group A. If you have blood type A, what you have is you have one allele. In this case, this is the big I with the big A attached. The I stands for immune factor. Immune factor. And this immune factor can either be an A, and that gives you blood type A, and it gives you blood type A because what it produces is these little triangles, these little carbohydrates on the surface of your red blood cell. That's what A produces. The other one you can be is B, And if you're B, it produces the little circle-shaped carbohydrates on the surface of your red blood cells. And then the second thing you can have is you can have a little I, which means the absence of an immune factor. So the big I means the immune factor is present, and it can either be A or B. And that's why we call it big I with a superscripted big A. And so if you have two of these, then you're blood type A. Or if you have one of those and the absence of the immune factor, then you're also blood type A. And then of course the same goes for B. Either you have two of the B's or one of the B's and the little I. And then for AB, and this is where the codominance bit comes in. In this case you have one of the little A's, one of the big A's up here and one of the big B's up there. And in that case, you produce both of these. So on the surface of your red blood cells, you have a mosaic of both of these carbohydrates. And then lastly, if you have none of these, then you're blood type O. It started out as blood type 0 because 0 was referring to 0 was the number of carbohydrates that are on the surface of your red blood cells. So that's denoted by little I, and that is the recessive. So here what you have is homozygous dominant for A, heterozygous for A. Homozygous dominant for B, heterozygous for B. Here you have heterozygous for AB. So this is the codominant part. Now what does this mean in terms of how this blood type actually expresses itself? Well, if somebody with blood type A receives blood type B, then they produce little antibodies, and the blood type B or AB is going to clot, kind of like curdled milk. That's what it looks like. But of course, if you get given your own blood type which has the right kind of markers, then you're fine. And if you're given blood type O, which doesn't have any markers, then you're also fine. O, in fact, is fine to give to anybody. So O is the universal donor. Because it doesn't have any of these little carbohydrates. Now the next thing to think about is what happens if you have blood type B. You have blood type B and somebody tries to give you blood type A. Well, that's not going to work because your body is going to react against blood type A and against blood type AB because that also has the little A bits on the top. So if you're blood type B, you can receive blood type O and blood type B. If you're blood type A and somebody gives you blood type, if you're blood type AB, and somebody gives you blood type A, then that's okay because you have these little triangles. So your body recognizes them. It's okay. Blood type B, same thing. Blood type O, there's nothing there, so that works. And so blood type AB is the universal recipient. So if you have blood type AB, then anybody can give you blood. And so this is how it rolls out. Blood type O can donate to everybody. Blood type AB can receive for everybody. Blood type O can only receive from other people with O. And that's how this works. Now, many of you know that there is also something with the plus and minus. There's some other thing called the rhesus factor. And so people may be, if you're A positive, right? Well, you have this additional gene, this rhesus factor, and that puts up little things as well. So the rhesus, you can be O positive, and that means you have the little rhesus markers. But that's a different gene altogether than the ABO one. But that's how your blood type is most commonly presented, A positive or B positive or whatever. So that's how the multiple alleles and codominants work with blood type. Now when you have blood type O, that's sort of to start out, that is the recessive phenotype. Recessive for everything. There's nothing on those blood cells. IA and IB, those are codominant. So if you have IA only, then you have blood type A. If you have IB only, then you are blood type B. If the two of them come together, that's the codominant version, and then you have blood type AB. So this is how the blood type works. It's kind of interesting. You have, a finite number. You have 3 possible alleles, yet you can produce 4 different blood types. And again, it comes from Mendelian genetics because of the way this assortment happens, independent assortment. If you remember that, that's how it works. The next one of these is Pleiotropy. Pleiotropy is also something that's not apparent from Mendelian genetics, but again, it doesn't have anything to do with Mendelian genetics being wrong. It has something to do with how your body responds to certain characteristics. And so pleiotropy can occur, for example, when you have one little glitch. So this would be the Mendelian thing, the abnormal hemoglobin blood protein, and how that is inherited. So that's your glitch. But then many other things could be affected by this. So if you have this defective kind of red blood cell, then what happens here? Well, it means that your red blood cells tend to break down more quickly. They clump more easily. They may damage parts of your spleen. And so there are multiple things that can go wrong as a consequence of this one little change. Now that little change, that underlies Mendelian rules, Mendelian principles. But all these other things, well, that's just over and beyond of what Mendelian genetics could predict. But Mendelian genetics still works based on this. It's just that there are all kinds of repercussions when you have this one condition. And then lastly, what happens if you have multiple genes whose effects, when combined, have an impact on a single feature? Human skin type is an example of this. Skin color in humans is actually produced by 3 genes. And if you studied this, you would see something like what's shown here on the right, where you have a parent generation and there's the 3 alleles, and you start with the homozygous recessive for each, and you made it to the homozygous dominant, and this ends up being a trihybrid cross. Now you don't have to know how to do this because there's just so many different little boxes, but the point is, when you look at this overall, you can come up with a distribution. And so there's your distribution. So you see that in each case there are relatively few people who have the very, very light skin type. You have very few people who have the very dark skin type, and sort of in the middle, that's where you find most of the people in terms of the skin type. And that's again predicated on genetics. Now if you were to do this experiment and you said, okay, let's come up with a color classification scheme, and we say, okay, this is very light skin, medium light skin, medium dark skin, very dark skin, and we somehow separated everybody in this room out according to these characteristics, we would probably be able to come up with some distribution like this. There are going to be very few people who have really light skin. There are going to be few people with really dark skin, but everything in the middle is sort of evenly distributed. And so that would give you polygenic inheritance. And so if you work through these 4 different problems, these 4 things that appeared not to be Mendelian in how they present themselves, you'd have to realize at the end they are indeed Mendelian. It's just that there are some contributing factors that people had not realized existed. Likewise, when it comes to the environment, there are some critical aspects of how the phenotype changes, and it has nothing to do with genetics at all. I mean, think about this for a minute. All the ways by which you can change your phenotype. You can cut your hair, you can put on makeup, you can dye your hair, you can get a tattoo, or you can, do some kind of like an earring or something like that. Whatever it is, those are all changes that you control, and yet they are not genetic in any way. Therefore, Mendelian genetics doesn't apply. Furthermore, even if you're twins, as these two ladies are, they are still environmental influences, even though their genetics are essentially the same. You know, one of them may fall and scrape her knee and the other one doesn't. That scar is not genetic. It's just an interaction with the environment. Or maybe when they're bicycling and one of them falls off and the other one doesn't. Right? One gets bitten by the dog and the other one doesn't. Whatever those kinds of changes are, they are entirely random. They are dictated by the environment, not by the underlying genetics, and that means that none of them, the scar, the hair color, the ear piercing, none of those are going to transfer into the next generation. So there are indeed some exceptions to Mendelian genetics when things aren't underlying genetics principles at all. When it doesn't have anything to do with genetics, of course, Mendelian genetics doesn't apply. That's what happens with these environmental influences.